PCR, that is PCR is polymerase chain reaction. This polymerase chain reaction, it was discovered by Caribulus in 1985. What is the application of this PCR? Basically, PCR is a kind of a reaction which is carried out in compact and compact and stable thermocyclers. These are the devices which have the ability of changing their temperature during the reaction is carried out. So, in a PCR reaction is generally carried out for amplification of DNA, that is, if you want to increase the copy number of DNA artificially, we use to carry out PCR. In general, if we want to increase the copy number of a DNA, this is the artificial method of increasing the copy number. For increasing the copy number by using a natural method, we use DNA cloning. So, talking about PCR, first of all we should have a look at the steps which are involved in a single PCR reaction. A single PCR reaction mainly consists of 3 steps. First is denaturation, second is annealing, and third is extension. This is the DNA molecule, target DNA molecule, which is to be amplified. In this, so first the target DNA molecule is taken. First step is denaturation, that is, this double stranded DNA is heated to 90 to 95 degrees Celsius. The 2 strands of the DNA, they get separated and we get single stranded DNA. Now second is annealing, around 40 to 60 degrees Celsius temperature. The primers which we used to design according to our requirement and according to our experiment, these primers, they anneal at the 3' end of the template DNA, target DNA. Why the primers they anneal at the 3' end only, why they do not anneal at the 5' end? Because the synthesis of DNA always takes place in 5' to 3' direction. So, when the primers they anneal at the 3' end, the synthesis of the new strand takes place in 5' to 3' direction. So, after the annealing of the primers to the template strand, third step is extension, which is carried out at 70 to 72 degrees Celsius with the help of Taq polymerase enzyme. We will discuss about what is Taq. Now, these primers, they are used as a support system for binding of the Taq polymerase enzyme. The Taq polymerase comes and binds here. It moves in 5' to 3' direction on both the strands, and we used to get 2 molecules of double stranded DNA at the end of a single PCR reaction with the starting of 1 molecule of double stranded DNA. Now, if we are carrying out a PCR reaction, what are the components which are to be added in a tube? So, for carrying out a single PCR reaction, in a 0.2 ml tube, we used to add following components. First is DNA sample, which is to be amplified, then Taq polymerase. Now, what is Taq polymerase? Taq is Thermus aquaticus. Thermus aquaticus. Why we have chosen this Taq polymerase? Because Taq is an enzyme which is taken from the bacteria Thermus aquaticus. This bacteria is found in hot regions. So, this enzyme has the tendency to remain stable even at high temperatures of 90 to 95 degrees Celsius. So, Taq polymerase plus DNTPs. DNTPs is deoxy Deoxynucleotide triphosphate, it includes like DATP, DGTP, DCTP and DTTP. What is the role? That while synthesis of the new strand, suppose A, G, C, T, T, if the template strand has A, so we need to incorporate T here. So this T, it will come from these DNTPs. Okay, so this is the role of DNTPs. So DNTPs plus primer plus magnesium ions plus water. So these are the components which are taken in a single reaction tube for carrying out a PCR reaction. The volume of the PCR tube is 0.2 ml. So talking about the application as we have already discussed, the main application of PCR is amplification of DNA. So this is a simple kind of a PCR reaction. Now if we have different kind of situations, then according to those situations there are different modifications of PCR. Like first is RT-PCR. What is RT-PCR? RT-PCR is reverse transcriptase PCR. Reverse transcriptase PCR, this kind of a PCR reaction is carried out when our sample is RNA. So if we want to increase the copy number of this mRNA molecule, suppose we have a experiment, and in that experiment we have planted 2 plants. In one of the plant we have given a stress condition, and now we want to see the effect of this stress. So in that case we need to study on mRNA, not on DNA. So in these cases we have to isolate RNA as the sample from the plant. Now this RNA, if we want to increase its copy number, we want to study on it further, then this mRNA is needed to be converted to DNA, and it then needed to be further amplified. So the steps are involved in this reaction are: this is the mRNA molecule having poly-A tail. In nature, the poly-A tail is present at the 3' end of mRNA. Now using reverse transcriptase enzyme, plus oligo DT as a primer, we synthesize first strand of DNA molecule. So this is mRNA and this is cDNA, that is the first strand. This is the 5' end, 3' end. Now we need to remove this mRNA from this hybrid, which is done with the help of enzyme RNase H. Now we get single stranded cDNA. Now in order to amplify this cDNA, we need to know its 3' end, so that we can design the primer corresponding to the 3' end, but we didn't know the sequence. So in order to overcome this situation, we will add We will take GTPs with the help of enzyme TDT, that is terminal deoxynucleotidal transferase. So at the 3' end, these GTPs they get added, and at the 5' end we have a poly T tail. So now we get a DNA sequence with whose both ends are known to us. So with the help of primers and with the help of PCR reactions, we will take oligo C and oligo A as primers, and we will continue to amplify this DNA by several PCR reactions. Oligo A plus oligo C, and we can obtain billions of copies of this mRNA in the form of DNA. This kind of a PCR reaction is generally used when our sample is RNA in nature. Thank you.